everyone, my name is Jeffrey Blake and I'm an incoming junior here at the University of Minnesota working in the Prince Lab. Over the course of the program, my project is focused on utilizing single nucleus RNA sequencing to analyze worsening RV function in a pig model of right-sided heart failure. This specific technology uses nuclei from individual heart cells to extract their RNA transcripts to be read into a digital library for analysis. This is important as it allows us to categorize populations of cells that may be present or missing in a disease state. What we found was that severe RVD was far more distinct than mild RVD when compared to control. This was due to a profound loss of cardiomyocytes, but a gradual gain in vascular and immune cell types. This led us to find that severe RVD pathology is mounted by shifts in perfusion, inflammation, and cardiomyocyte metabolism. Thank you, everyone, and that was my project.
Hi, my name is Catherine Bryan, and I'm in the OGLE lab. Hypertrophic cardiomyopathy, or HCM, is the most common genetic cause of heart disease. It leads to many symptoms and in some cases heart failure, but we still don't fully understand what drives the condition. For my project, we used stem cell-derived heart tissues with an HCM-linked mutation to model the disease. I compared these mutant tissues with healthy controls and found that key cilia-related proteins were significantly downregulated in the HCM tissues. Through further protein analysis and cardiac cell imaging, we confirmed that HCM cells are missing tiny antenna-like organelles called primary cilia. These cilia play an important role in cell signaling pathways. Without them, a protective pathway called sonic hedgehog is turned off, and a fibrosis-promoting pathway called TGF-beta becomes overactive. These findings suggest that the loss of primary cilia is a key trigger of fibrosis in HCM. Future studies will expand on primary cilia's role in HCM and explore how addressing degraded cilia might offer new treatment approaches.
Hi, my name is Shlok Jani, and I'm a LHI summer scholar working in the Kamdar lab. This summer, I studied how heart cells handle calcium, an especially vital aspect of heart function, given that calcium release and storage is responsible for the heart's contraction and relaxation every heartbeat. In healthy heart cells, calcium is tightly regulated. A key player in this process is a protein called SERCA, which acts as a pump that stores calcium inside heart cells. However, in certain diseases like Duchenne muscular dystrophy, DMD, SERCA's function is impaired, leading to calcium buildup and irregular heartbeat. To see if we could improve SERCA function in healthy heart cells, we tested a therapy where we delivered extra SERCA using a virus into human stem cell-derived heart cells. We found that our therapy boosted SERCA levels, improved calcium storage, and even sped up how quickly the heart cells release calcium. Our research indicates that SERCA gene therapy may be a potential treatment for patients suffering from heart diseases characterized by calcium dysregulation, opening the door for further research into developing treatments for genetic heart diseases like DMD. Thank you.
Hey everyone, my name is David John and I'm a current student in Dr. Michael Kaiba's lab here at the U. One of the diseases that my lab studies is FSHD, facioscapulohumeral muscular dystrophy, which is the third most common inherited muscle disease in the world. And what's interesting about this disease is it's primarily been studied in the lens of the muscle cell. FSHD is caused by the unusual expression of this gene and protein DUX4, which is normally silenced in healthy muscle cells. But when DUX4 is expressed, it leads to a cascade of cytotoxic effects in the cell that is ultimately detrimental and leads to the death of the cell, which is why in FSHD patients, muscle atrophy is one of the key characteristics. But what we've been doing in the Kaiba lab is exploring non-muscle lineages in things like neurons, for example. So what I've been doing this summer is I've been taking inducible pluripotent stem cells and differentiating these stem cells into mature motor neurons. I've been collecting samples for things like RNA sequencing, immunostaining, as well as qPCR, and I've been looking for DUX4 expression throughout the whole timeline, as well as some of those target genes which DUX4 affects. And we've got some really interesting findings by looking at neurons, which hasn't been done in a lab ever before in regards to FSHD. So these findings could change the whole landscape of what FSHD is, as now we show the potential that there might be some component with the central nervous system in some way. And I'm really grateful to be a part of this lab and to experience groundbreaking research where it really takes place, which is in the lab. So thank you.
Hi, I'm Madeline, and I have the privilege of working in the Dudley Lab. SGLT2 inhibitors are drugs developed to help patients with type 2 diabetes regulate their glucose levels, but they also have cardioprotective effects, even on non-diabetic patients. Since many patients with diabetes have low magnesium levels and high levels of inflammation and oxidative stress, we set out to determine whether an SGLT2 inhibitor, dapagliflozin, plays its cardioprotective role via affecting these mechanisms. We first set up a low magnesium mouse and cell model and treated these models with dapagliflozin. Using multiple technologies, we found that dapagliflozin impacts cardiomyocytes and improves diastolic dysfunction by raising magnesium levels. This reduces the expression of a magnesium transporter with a kinase function, TRPM7. Suppression of TRPM7 kinase reduces oxidative stress and inflammation, which contribute to diastolic dysfunction. These findings are important to better understand how dapagliflozin and probably other SGLT2 inhibitors can be used to treat diastolic dysfunction. Thank you.
Hi, my name is Violet Johnston, and I'm a proud part of the Townsend Lab. This summer, I was part of two projects. First, I helped revise our Langendorf setup, which allows for ex vivo perfusion experiments. And two, I studied DMD, known as Duchenne muscular dystrophy, within mouse hearts. DMD is a genetic disease that causes muscle dystrophy, so muscle to waste away, including in the heart. Usually, people have a protein called dystrophin, which acts as a stabilizer or protectant for the heart muscle cells, but DMD patients don't have this protein. So scientists have identified a possible treatment through gene therapy by creating this much smaller protein called microdystrophin. This summer, I looked at using microdystrophin as a possible way to help restore cardiac health in some of these patients. We took a bunch of mice, we gave them this cardiac strain that induced some of similar symptoms to what DMD patients would go through, and then we tested for expression of microdystrophin levels before and after treatments.
Hello, my name is Dylan Kim, and I'm a rising senior at Westwood High School. This summer, I had the extraordinary opportunity to be mentored by Dr. Bosniakovsky while working on the characterization of the IDUX mouse, which is a novel murine model that my lab generated to study facioscapulohumeral muscular dystrophy, or FSHD. FSHD is a common and progressive myopathy that is caused by the failure to silence the DUX4 gene. The key feature of the IDUX mouse, however, is a doxycycline-inducible mouse DUX gene, the murine paralog of human DUX4, expressible specifically in skeletal myofibers. To our knowledge, this is the first FSHD model to implement mouse DUX, and upon histological analysis, we discovered that the IDUX mouse recapitulated key aspects of FSHD pathology, including alterations in myofiber size, fibrotic ECM deposition, and mononuclear cell infiltration. These findings are important because they demonstrate that mouse DUX produces similar effects as its human counterpart, demonstrating that this model may be used in the future to further study FSHD.
Hi, my name is Vivian Krzyzewski-Meyer, and I'm a rising senior at De La Salle High School. This summer, I've been interning in the Prisco lab, where we've been studying the disease pulmonary arterial hypertension, or PAH. PAH is a rare and lethal cardiovascular disease, and it leads to right ventricular failure. Right ventricular failure is the primary predictor of mortality outcomes in the disease, but there are no current treatments that target the failing RV. There are trillions of tiny microbes in our gut, but we realized that the role of the gut microbiome, or the fungal species, had been severely under-researched in PAH. With our study, we wanted to see whether or not the supplementation of the fungus Bloomeria would affect PAH severity and RV function in the disease. With directly supplementing a rodent preclinical PAH model, we had found that it enhanced RV function for the rats. This shows that it would be a novel approach in the future for directly targeting a failing right ventricle in PAH.
So hi, my name is Belle and I'm in the Leo lab. This summer I've been doing research on the comparison mitochondrial function in the left or right ventricles and in young and aged mice. So why are we studying mitochondria? Well, mitochondrial dysfunction causes negative impact on cardiac function and eventually heart failure. And in this experiment, we tested mitochondria from isolating it from the right and left ventricles in mice and performed experiments such as collecting oxygen consumption rate, ATP production, and calcium uptake. And in these three experiments, we found higher values in young mice compared to the old. And it's also interesting to see that there were higher values, tens of higher values in the right ventricle compared to the left. And so future research would definitely include increasing the number of samples and seeing if we would observe these results further.
My name is Rachel Proniainen, and this summer I was in the Van Berlo lab working on PDK4's role in cardiomyocyte maturation. So cardiomyocyte maturation is important because stem cell-derived cardiomyocytes are impossible to mature outside of the body, and they remain permanently immature. So I focused on metabolic maturation using PDK4, which inhibits a complex that transitions pyruvate from glycolysis into acetyl-CoA for the TCA cycle. And so by upregulating PDK4 and blocking that complex's activity, the body is forced to use fatty acid oxidation as a source of acetyl-CoA for the TCA cycle instead of using glycolysis, glycolysis is a more immature form of metabolic use, and then fatty acid oxidation is considered a more mature form. So I investigated PDK4 in two ways. First, I isolated neonatal cardiomyocytes, and then I treated them with single interfering RNA to knock down PDK4 levels. Then I tested the knockdown and known maturation markers in cardiomyocytes using qPCR. Then I also used 4 different developmental time points with mouse hearts and then stained them to see how PDK4 expression changes over time. And what I found is that for the sRNA, unfortunately, my data wasn't significant, but there was a general trend that with less PDK4 expression, the cells do seem to be less mature. And then for the staining, I found that there's a strong nuclear presence for PDK4 throughout development, which hints at a new function that's not yet been described for PDK4. Thank you.